DNA replication occurs in the S phase of interphase. During this process, DNA is replicated using a semi-conservative model, in which each daughter cell has one old strand and one new strand. This creates two new strands of DNA. First, a replication bubble is formed at special sites known as the origins of replication. At the end of the replication bubble, a Y-shaped region called the replication fork separates the two parental strands of DNA. The enzymes that unwind the DNA and separate it are the helicases. Also, the single-strand binding protein, which binds to the unpaired DNA strand, stabilizes it.
The unwound strands of DNA can now serve as a template for the synthesis of complementary DNA strands. The initial nucleotide chain that is produced during DNA synthesis is actually a short chain of RNA. The small chain of RNA is synthesized by an enzyme known as the RNA primer that starts off the complementary DNA strands. The new DNA starts at the 3' end of the RNA primer.
DNA polymerase are the ones that catalyze the synthesis of new DNA by adding to the pre-existing RNA primer. Most DNA polymerase cannot start without the RNA primer. The DNA polymerase continues to elongate the chain by adding new nucleotides at the 3' end. The rate of elongation is about 50 nucleotides per second in a human cell.
There are two types of strands during DNA replication. The first is the leading strand, which goes from 5' to 3', and the other strand is the lagging strand, which goes from 3' to 5'. Since the RNA primer and the DNA polymerase can only go from 5' to 3', the lagging strand makes fragments of DNA known as Okazaki fragments. DNA polymerase I adds DNA nucleotides in place of the RNA nucleotides to the 3' end of each Okazaki fragment. DNA ligase then joins the sugar phosphate backbones together and creates a continuous strand of DNA.